So, generally in plants linear growth it is due to apical and intercalary meristems. So, linear growth that is due to primary meristems. What means? Apical and intercalary meristems. But next one, that growth in girth, it means increase in thickness or diameter or girth. It takes place by lateral meristems. So, increase in thickness by lateral meristems is called secondary growth. Generally, lateral meristems are intrafascicular cambium, then interfascicular cambium, and also car cambium. These are lateral meristems. Then point is, among these three, intrafascicular is a primary meristem, inter and a car cambium. These are secondary meristems. It means both primary, secondary meristems. Both meristems participate in secondary growth. Then point, generally secondary growth absent in pteridophytes. Secondary growth absent in pteridophytes. But some pteridophytes like 𝗖𝗍𝗍𝗪, it shows secondary growth. Then secondary growth common in gymnosperms and dicots. Gymnosperms and dicots, secondary growth takes place. And secondary growth absent in monocots. But some monocots like 𝗂𝗏 and 𝗓𝗏, they shows secondary growth. Then in which parts actually secondary growth takes place means, secondary growth occur in both the stems and roots. Secondary growth occur in which parts means stems and roots. Then first one, secondary growth in stems. That too, in stems, secondary growth occur in two parts. One is within stele. It is called intrastellar secondary growth. Then second one, outside the stele. This outside the stele is called extrastellar secondary growth. Which cambium responsible for intrastellar secondary growth means vascular cambium. Vascular cambium responsible for that intrastellar secondary growth. So, vascular cambium is called stellar cambium. Then, which cambium responsible for extrastellar secondary growth means car cambium. So, car cambium is responsible for extrastellar secondary growth. And this extrastellar secondary growth which occur in cortex. So, car cambium is called cortical cambium. Now, detailed description about intrastellar secondary growth. First, that a steel in a dicot stem is called U steel. Steel in dicot stem called a U steel. U steel means ring of vascular bundles. If ring of vascular bundles present, called U steel. Then in each vascular bundles, first primary vascular tissues. This primary vascular tissues means primary phloem and primary xylem. Phloem and xylem. These primary vascular tissues produce from procambium. Primary vascular tissues, they produce from procambium. After formation of primary vascular tissues, some amount of procambium persists in between this primary xylem and phloem. That is called intrafascicular, or it is called fascicular cambium. Intrafascicular or fascicular cambium. So intrafascicular or fascicular cambium is primary meristem because produced by differentiation. So it is primary meristem. Then in dicot stem, there is medulla. And some of the medullary cells extend in between vascular bundles, rightly called medullary rays. These are primary medullary rays. Generally, these primary medullary rays are parenchymatous. Then, this primary medullary ray by dedifferentiation produces cambium. Primary medullary rays by dedifferentiation they produce cambium. That cambium present in between vascular bundles is called interfascicular cambium. This interfascicular cambium produced from medullary rays by dedifferentiation. So it is secondary. So point is, cambium within vascular bundle is intrafascicular or fascicular, which is primary. Cambium in between vascular bundles is interfascicular. That interfascicular is secondary because produced by dedifferentiation from medullary rays. Then this interfascicular cambial stripes joins with this intrafascicular stripes and forms new cambial ring. That new cambial ring is called vascular cambium. So vascular cambium is lateral meristem, but it is partly primary, that is intrafascicular or fascicular, and partly secondary, that is what? Interfascicular. So point is, generally lateral meristems are secondary. They participate in secondary growth. But here only one, intrafascicular cambium, that is primary lateral meristem, participate in secondary growth. So, based on that we can say all lateral meristems secondary, wrong. There is one primary lateral meristem participate in secondary growth. So, primary lateral meristem which participate in secondary growth is intrafascicular or fascicular cambium. Then once cambial ring is formed, this cambial ring undergoes periclinal division. Cambial ring undergoes periclinal division and produce secondary phloem outside and secondary xylem inside. So, it is secondary phloem, then it is secondary xylem. So, point outside this vascular cambium produce secondary phloem, inside this vascular cambium produce secondary xylem. It means vascular cambium centrifugally outside produce secondary phloem, centripetally inside it produce secondary xylem. Here point is it produce more amount of secondary xylem than that secondary phloem. In normal secondary growth, more amount of secondary xylem is formed than that secondary phloem. So, in that way, once vascular cambium is formed, every year continuously it produces secondary xylem. Every year continuously it produces that a secondary xylem, so that pressure developed on that a secondary phloem and even primary phloem. So, the secondary and primary phloem get crushed and removed. That is why large amount of secondary xylem is formed by this secondary growth. Then rightly this vascular cambium, it contains two types of cells. One is called fusiform initials and second one is called ray initials. Fusiform initials, they gives rise to secondary vascular tissues like secondary xylem and phloem. Ray initials, they gives rise to secondary medullary rays. Actually, these are primary medullary rays. This secondary medullary rays joins with primary medullary rays and they participate in that lateral conduction or radial conduction. And part of medullary ray present in phloem called phloem rays or bast rays. Part of medullary rays present in xylem called xylem or wood rays. Your point is secondary xylem is called wood, then secondary phloem is called bast. Then point newly formed secondary phloem position. That newly formed secondary phloem position is just outside vascular cambium. Newly formed secondary xylem position just inside vascular cambium. This is about secondary growth. Then in old dicot stem, in that centrally placed wood, in old dicot stem, centrally placed wood, in that their vessels or tracheids are filled with ergastic substance like resins, tannins, gums, essential oils and aromatic compounds. Due to deposition of all these compounds, that is dark in color generally it lost that its main function, that is what means conduction, and particularly vessels of this wood is blocked by balloon-like structures. These balloon-like structures are called 𝗕𝗏𝗧𝗔, and 𝗕𝗏𝗧𝗔 also called xylem plugs. So due to formation of 𝗕𝗏𝗧𝗔 or xylem plugs, they are not useful for conduction. So 𝗕𝗏𝗧𝗔 stop that entry of water, and even it shows some advantages formation of the 𝗕𝗏𝗧𝗔. How means it prevents that movement of or entry of microorganisms or pathogens. And this type of wood, which is dark in color, then it is highly hard, durable, resistant to pest, and such wood is called heartwood, and it is highly durable. Hence this heartwood is also called that duramen. Heartwood is also called duramen. So if heartwood is not useful for conduction, that plant will die. So how this conduction takes place? How this conduction takes place means immediately vascular cambium. This vascular cambium, it produces new wood. That new wood is peripherally placed. Generally it is yellow colored. Such wood is called sapwood, and this sapwood is also called alburnum, and this alburnum or sapwood help in conduction. So in old dicot stem, centrally placed wood is heartwood, also called duramen, and a newly formed wood which is peripherally placed, that wood is called sapwood or alburnum. Then gradually the sapwood is converted into heartwood. How means? Due to formation of 𝗕𝗏𝗧𝗔. So due to formation of 𝗕𝗏𝗧𝗔, gradually that sapwood is converted into that heartwood. Then now types of wood. In angiosperms, wood contains vessels, rightly called porous wood. Angiosperms. So angiosperms, wood is porous. Porous means it contains vessels. In gymnosperms, that wood is without vessels, rightly called non-porous wood. Gymnosperms vessels absent except neatum. In neatum, that vessels are present. So generally porous wood means if vessels present, called a porous. If vessels absent, called a non-porous wood. Then this porous wood is of two types. One is called diffuse, and second one is called ring porous wood. Diffuse and a ring porous wood. Diffuse means these xylem vessels throughout the wood, that the xylem vessels are uniformly distributed. That is called diffuse. Example for diffuse: betula. Betula, common name, that 𝗵𝗁𝗆. Then next one, ring porous wood. Ring porous wood means that vessels are not uniformly distributed. More vessels present in early wood than late wood, called ring porous wood. Example for ring porous wood, that quercus. These are types of wood. Particularly in gymnosperms, there are another two types of wood is present. One is called monoxylic and second one is called pycnoxylic. Monoxylic means that the xylem is soft. Monoxylic, xylem is soft because it contains more amount of parenchyma. So, called monoxylic. Example for monoxylic, cycas. The next one, pycnoxylic. Pycnoxylic means that xylem is hard because pycnoxylic means less amount of xylem parenchyma is present. The xylem parenchyma is compactly arranged, so it is called pycnoxylic. Example, pinus. Example is pinus. Then, importance of some wood, that wood of 𝗕𝗏𝗃𝗃. 𝗕𝗏𝗃𝗃 means red sanders. 𝗕𝗏𝗃𝗃, red sanders, wood used to make musical instruments. 𝗕𝗏𝗃𝗃. Common name is red sanders. Then wood of 𝗪𝗏 𝗪𝗏, rosewood. In rosewood, this wood used to make furniture. Then that essential oil, that sandalwood oil. Sandalwood oil is obtained from this heartwood of that sandalwood. These are some of the importance of wood. Next one, annual rings. Annual rings. Here, chemical activity. Chemical activity depends on the season. It means chemical activity, it shows seasonal variations. Particularly in temperate plants, that in spring season, in temperate plants, in spring season, new leaves are formed. So, temperate plants, in spring season, new leaves are formed. Then, new leaves means it shows high metabolic activity. It means it need more amount of water. So, in spring season, activity of vascular cambium increased. This vascular cambium produce large amount of vessels with a wide lumen. Such wood is called spring wood, or it is called early wood. Then in autumn season, that leaves fall off. Leaves fall off means metabolic activity is decreased. Metabolic activity is decreased, the tree does not require much water. So, activity of vascular cambium also decreases. And this vascular cambium produce less amount of vessel with narrow lumen. Such wood is called late wood, or it is called autumn wood. So, for formation of one spring and one autumn wood, it takes 1 year. So, this spring wood formed inner side, it is lighter in color. Then autumn wood, which is formed outside the spring, this autumn wood is darker in color. Then the spring and autumn wood, for formation of these two wood, it takes 1 year, rightly called annual rings or also called growth rings. If annual rings are clear, called growth rings. So in temperate plants, annual rings are clear, so called growth rings. What is the use of these annual rings? These annual rings, they help in estimating the age of tree. That such science is called dendrochronology. Generally, annual rings help in that dendrochronology, but one exceptional, that a 𝗧-𝗪𝗃 plant. In 𝗧-𝗪𝗃, that age can be estimated by that leaf basis, and in 𝗧-𝗪𝗃, for formation of leaf, it takes 1 year. After that, entire leaf is fall off. Only leaf base is persistent. All that leaf bases, they forms armor of sheath over the stem, and it help in that estimating age of tree. By counting these annual rings, that age of that one of the old tree, 𝗧𝗦𝗔𝗧, its age can be estimated as 3,500 years. So annual rings are clearly formed in temperate plants. If clearly formed, called growth rings. In tropical countries like India, there is no much seasonal variation. So annual rings are formed, but that annual rings are not clear. So if annual rings are not clear, rightly called growth marks. Sometimes annual rings are formed due to heavy leaf fall or hormonal changes. Such type of annual rings are called pseudo annual rings. That is why annual rings are not exact or accurate to estimate age of tree. This is about intrastellar secondary growth. Next one, extrastellar secondary growth. Extrastellar secondary growth. Then your point is at center of stem medulla. Medulla is surrounded by primary xylem. Primary xylem is by secondary xylem. Secondary xylem surrounded by vascular cambium. Vascular cambium is by secondary phloem. Secondary phloem is by primary phloem. Primary phloem. From center to outside, medulla, primary xylem, secondary xylem, vascular cambium, then that secondary phloem, then primary phloem. So when inside, due to the activity of vascular cambium, when inside still, if secondary tissues are formed, that pressure developed on outer side. So particularly outer protective layers like epidermis and its underlying tissue like a cortex destroyed. So these protective layers are replaced by new protective layer. Such new protective layer is called what means periderm. So how this periderm is formed? Now outside primary phloem, endodermis, pericycle has the old stem. These tissues are replaced, but there is primary cortex. in most of the cases, this primary cortex undergoes dedifferentiation. It undergoes dedifferentiation and it produces cork cambium. This cork cambium is also called phyllogen. So in most of the cases, this cork cambium or phyllogen, it is formed from primary cortex by dedifferentiation. See, in dicot stem, vascular cambium is partly primary, partly secondary. Partly primary is intrafascicular, partly secondary is interfascicular. But total cork cambium is a secondary. Then point, generally cork cambium is produced from primary cortex. Then in some plants, cork cambium is produced from epidermis. Example, salix. In some plants, it is produced from cortex. Example, clematis. Cortex, clematis, epidermis, salix. Then, after formation of cork cambium, this cork cambium undergoes periclinal division. It undergoes periclinal division and it produces cells on both sides. Outside cells are called phylum or cork. Inside it produces secondary cortex. Inside it produces secondary cortex. This secondary cortex is called phylloderm. Secondary cortex is called phylloderm. So point is cork or phylum, cork cambium or phyllogen, secondary cortex or phylloderm, together called periderm. So once again, periderm means cork or phylum, cork cambium or phyllogen, secondary cortex or phylloderm, together called that a periderm. So old dicot stem, protective layer is that a periderm. Now first one, cork. Cork cells generally develop from cork cambium outside. In young stage, cork cells are living and cellulosic. But after maturity, due to deposition of suberin, the cells become dead. And because of suberin, these cork cells impermeable to water and gases. They are compressible, lighter in weight. These are thermal insulating properties. That is why this cork tissue used to prevent that a fire, and a commercial cork is obtained from that a Quercus 𝗂𝗏, common name Indian oak tree. And Robert Hooke first discovered that a dead cell, that is cork cell, from which plant means? From Quercus 𝗂𝗏. He discovered that a cell but he studied structure of cell wall and he gave the term that a cell. And cork cells generally rectangular, closely arranged without any intercellular spaces. Then next one, secondary cortex. Cortex means parenchymatous. So here secondary cortex, that cells are spherical or oval with intercellular spaces. And this secondary cortex, it contains what means chloroplast. So it help in photosynthesis. So question, function of secondary cortex, photosynthesis. Actual question, assimilatory tissue produced from cork cambium is what means that a secondary cortex or phylloderm. Secondary cortex or phylloderm. Here point, this secondary cortex or phylloderm, it is photosynthetic. So it required gases. It present inner side to that cork cambium. Outside cork cambium there is cork. Cork is made up of suberin. So it is impermeable to water and gases. But so how this secondary cortex receives that gases means because of that this cork cambium. This cork cambium produce non-suberized parenchymatous cells. These non-suberized parenchymatous cells produced from cork cambium are called complementary cells. Called what? Complementary cells. And due to formation of complementary cells, that a pressure developed on that a cork tissue, then this cork tissue splits and open called lenticels. So in an old dicot stem, transpiration or respiration takes place through that lenticels. Through lenticels. This is extrastellar secondary growth. Now point, what is bark? Bark, there is technical and non-technical term. Technically, bark is outside cork cambium, and non-technically, all tissues present outside vascular cambium forms bark. It means bark includes both the living and dead tissues. Bark includes both the living and dead tissues. Then here living and dead tissues. Generally after secondary growth, that primary phloem get crushed and removed. So which layer present outside vascular cambium means that a secondary phloem. So living bark includes secondary phloem, primary cortex, then a secondary cortex. And dead bark, whatever tissues present outside cork cambium, that is what? Dead bark. So living bark present in between vascular cambium and a cork cambium, living bark. So innermost living bark is secondary phloem. Innermost living bark is secondary phloem. Outermost living bark is that a cork cambium. Then whatever tissues present outside cork cambium, all the tissues forms dead bark. Then bark is of two types. One is scale bark, but bark is removed as scale, called a scale bark. Example for scale bark. One is CDM and the second one eucalyptus. Then next one is ring bark. That bark removed as ring, rightly called ring bark. Example for ring bark. One is mango and second one is neem. Then quinine alkaloid. Quinine alkaloid is obtained from bark of cinchona, used to cure malaria. Then spicy bark is from that Cinnamomum zeylanicum. Cinnamomum zeylanicum. This spice you called dalchini. It is about extrastellar secondary growth. Then tissues from inside to outside. In extrastellar means primary cortex, secondary cortex, cambium, cork. Total from inside to outside. First medulla, then primary xylem, then vascular cambium, then secondary phloem. So first medulla, generally medulla and primary xylem crushed, but it present as thin layers. Medulla, primary xylem, secondary xylem, then vascular cambium, outside vascular cambium, secondary phloem, generally primary phloem crushed or patches of primary phloem present, then outside primary phloem, primary cortex, outside primary cortex, secondary cortex, outside secondary cortex, that cork, cambium, then cork, then cork. So, in dicot stem, secondary growth occur in stele and also cortex. Stele means intrastellar. Which cambium? Vascular cambium. In dicot stem, vascular cambium is partly primary. That is what? Intrafascicular. Partly secondary. That is what? Interfascicular. And outside the stele, in cortex, which tissue responsible for secondary growth in cortex means cork cambium. So, cortical cambium is cork cambium. In dicot stem, cork cambium totally secondary in origin. Generally developed from primary cortex. By what? De-differentiation. Why? Primary cortex is parenchymatous. This is secondary growth in dicot stem. Next one, secondary growth in a dicot root. In dicot root. So, in dicot root also secondary growth occur in two places. Within stele called intrastellar, outside the stele called extrastellar secondary growth. Then point, within stele, which tissue responsible for secondary growth? Vascular cambium. Vascular cambium. In dicot root, starting stage, vascular cambium is absent. Starting stage, vascular cambium is absent. So that vascular bundles are closed. Later stage, this vascular cambium along with vascular cambium, even 𝗧𝗃𝗵𝗵 is formed. So, in later stage, that is secondary growth takes place. How this secondary growth occurs? With a simple example I will tell. Generally in dicot root, limited vascular bundles present, monarch, one xylem and phloem, and the most common tetrarch. Tetrarch means 4 xylem and phloem bundles present. This is xylem, primary xylem, then primary phloem. They are separate, alternate, present on different radii, called what? Radial vascular bundle. Radial vascular bundles. So, this is primary phloem. Actually, this xylem and phloem connected by parenchyma. This parenchyma is called conjunctive tissue. So, this is conjunctive tissue. Here it is what? Pericycle. So, first what happens means the parenchyma present below the primary phloem means conjunctive tissue. This conjunctive tissue undergoes dedifferentiation and it produces 4 strips of this vascular cambium like this. This pericycle undergoes dedifferentiation. Later, pericycle undergoes dedifferentiation and produce cambial stripes. Actually, where pericycle undergoes dedifferentiation means the pericycle present above the protoxylem. The pericycle present above the protoxylem undergoes dedifferentiation. Then it produces cambial stripes. So, this outer cambial stripes joins with inner cambial stripes and thus new vascular cambium is formed or a circle of vascular cambium is formed, but this vascular cambium is wavy. So, this is formation of vascular cambium in dicot root. Same, after formation of vascular cambium, this vascular cambium, it produces secondary phloem outside, then this secondary xylem inside. Secondary phloem outside and secondary xylem inside. So, it is secondary growth in dicot root, that too intrastellar. Then extrastellar secondary growth also occur in dicot root, which is similar to dicot stem. Like a dicot stem, even in a dicot root, due to formation of secondary xylem and a secondary phloem, that a pressure developed on outer tissues. So, outer tissues like epidermis and its underlying tissues, that outer cortical cells are destroyed, they are removed by new protective layer. That new protective layer is called periderm. That periderm is formed from that cork cambium, cork and also secondary cortex. Secondary cortex. So, it is intrastellar and extrastellar secondary growth in dicot root. So, next one, abnormal or anomalous secondary growth. Normal secondary growth. Normal secondary growth more amount of secondary xylem is formed and less amount of secondary phloem is formed. That is called normal secondary growth. Other than that, it is called anomalous or abnormal secondary growth. Here, dicots showing abnormal or anomalous secondary growth. One is bignonia, bauhinia, and bommeria. These are dicots showing anomalous secondary growth. Then monocots showing abnormal or anomalous secondary growth. Dracaena, 𝗂𝗏, even 𝗂𝗵𝗋 shows anomalous secondary growth. Then gymnosperms, even some gymnosperms, they shows abnormal or anomalous secondary growth. For this, one is 𝗬𝗏 and second one is 𝗧𝗏𝗃. Then generally anomalous secondary growth common in woody climbers. These woody climbers are called lions or lionas. Lions or lionas. Example for these lions or lionas, one is 𝗕𝗏𝗚𝗨, that is gymnosperm. Even some storage roots, shows abnormal or anomalous secondary growth. For this, carrot and also beetroot, they shows anomalous secondary growth. Simple description: 𝗧𝗏𝗃, in a 𝗧𝗏𝗃 stem, actually 𝗧𝗏𝗃 stem, many rings of vascular cambium are formed. So many rings of vascular cambium are formed, so many rings of xylem is formed. That is called polyxylic. And generally, this xylem contains large amount of parenchyma. So that xylem is soft, called polyxylic and monoxylic in a 𝗧𝗏𝗃 stem. Then 𝗧𝗏𝗃 root, two types: normal and modified adventitious roots. Modified adventitious roots are called coralloid roots. Modified adventitious roots called coralloid roots. These coralloid root shows only extrastellar secondary growth and it contains lenticels, that lenticels present on periderm. Then next one, in a lions means woody climbers. They take the support of nearby plant. So if during secondary growth, if more xylem is formed, it cannot climb. That is why during secondary growth, it produces more amount of secondary phloem. So it is called abnormal or anomalous secondary growth in lions or lionas. 𝗕𝗏𝗚𝗨 example. Then storage roots like carrot and beetroot. At the time of secondary growth, they produce large amount of secondary parenchyma for storage, hence called abnormal or anomalous secondary growth. So, these are examples for abnormal or anomalous secondary growth. So, finally, dicot root, dicot stem, both shows secondary growth. That too, two types of secondary growth, intrastellar and extrastellar, with a small differences. In dicot stem, vascular cambium is partly primary, that is intrafascicular, partly secondary, that is interfascicular. Both are joined to form vascular cambium. So, in dicot stem, vascular cambium partly primary, partly secondary. And in dicot stem, cork cambium is totally secondary. And that cork cambium generally develop from that primary cortex. Then dicot root. In dicot root, both vascular and cork cambium, both are totally secondary. Both are developed from that pericycle and conjunctive tissue. Primary lateral meristem developing three types of secondary tissues is fascicular, interfascicular, pericycle and endodermis. Answer is fascicular, because fascicular cambium is actually primary lateral meristem. It joins with interfascicular cambium and forms vascular cambium. That vascular cambium contains two types of cells, fusiform and reinitials. Fusiform and reinitials. So, they produce secondary xylem, secondary phloem, secondary medullary rays. So, three types of secondary tissues formed from this fascicular cambium. The next one, innermost living bark. Just now I discussed bark, living bark present in between vascular cambium and a cork cambium, and a dead bark present outside cork cambium. So, just outside vascular cambium, secondary phloem is present. So, innermost layer of living bark, secondary phloem or bast. The next question, sapwood is also called. So, another name for sapwood is alburnum. Then, tissue present in lenticels is epithem, epithelium, complementary and all. Here, epithem means colorless parenchyma. Epithem is colorless parenchyma which is present in that 𝗳𝗿𝗄𝗏. Epithelium, it is secretory layer present in schizogenous cavity. So, complementary cells present in lenticels. Then phylogen means a cork cambium and a vascular cambium. Both are secondary in origin. In answer, dicot root. How means? In dicot root, vascular cambium, cork cambium not present from starting stage. They developed after certain age of the root by what means? De-differentiation. So, phylogeny and vascular cambium both are secondary in dicot root. In dicot stem, phylogeny is totally secondary, but vascular cambium is partly primary and partly secondary. So, answer is dicot